Okay, this is your class questions book for component 2, and this is question number 4, the really interesting one on the SRY gene. Well worth looking up, really interesting, especially of interest to athletes, and you'll see why when you look it up. So, the SRY gene on the Y chromosome codes a protein involved in the development of testicular tissue and producing male hormones. So here we've got a map of the X and Y chromosomes of a normal male. So here's this SRY gene at the top. This is his X chromosome, then this is his Y chromosome. And our first thing is to identify structures A and B. So B, I'm going to do first because that's where they're joined together, and I know that is called a centromere. And then A is going to be a chromatid. So, during meiosis, 4 different sperm can be produced following sex chromosomes. So we've got sperm 1 with one arm of the X chromosome in, and sperm 2 has got that, an SRY gene. So this, we've got a bit of a transfer from the Y chromosome to the X chromosome. How does that happen? That's the idea of crossing over. We've got a sperm now that doesn't have the SRY gene on, and we've got one that is as it was. Okay, so these two have been altered. Explain how the gene can be transferred from Y to X. The only way of a gene getting from one chromosome to another is by crossing over. Transfer between chromatids occurs in prophase 1 during crossing over. And you could of course describe chiasmata formation, and you could use that lovely term synapsis. No need. So, another explain. Explain how we get 4 different and haploid sperm at the end of meiosis. So, in meiosis, so I'm going to explain where we get the 4 cells from. In meiosis, there are 2 divisions. In the first, the homologs separate into 2 cells, and in the second, the chromatids separate. One into each of the 4 cells. So that explains the haploid bit. I probably could have done it in much shorter words. And then they are all different because of crossing over and random assortment. So here we've got a picture that shows crossing over. We've talked about crossing over. We've talked about crossing over again. I'm thinking that this is pretty much a question about crossing over now. The presence of the SRY gene was used to check the gender of athletes. 8 out of over 3,000, so 0.23%, had the SRY gene. With reference to the diagrams given, so that means I want to refer back over here, and my knowledge of meiosis, suggest why this figure is so low. So, what we're saying is that some females inherit from their dad this one with the SRY gene, this chromosome here with the SRY gene, and from their mum presumably one that doesn't have it. So they've got the SRY gene coding for male hormones and testicular tissue even though they're females. So why is that not happening all the time? Remember that this has been crossing over. So this whole question is scaffolded to make you be thinking, crossing over, what do I know about crossing over? Why do we not make all of those for sperm all the time? So why would it not be 1/4, 1/4, 1/4, and then 1/4 of the female athletes instead of 0.23%? Well, if we look at the chromosomes, they're completely blinking different sizes. So although they're a pair that need separating at meiosis, they're such different sizes that it's really unlikely that they are going to do this crossing over thing. So, crossing over is less likely in sex chromosomes because they are different sizes. The Y chromosome is smaller, and now I have referenced my diagram. And you could put crossing over is rare, more difficult, has a lower probability, any of those ways of putting it. But it is just less likely just because they're totally different sizes.